of all the things we do, to have the greatest impact on public health. It also is an absolutely stupendous science project. So there, and that really, if you're in this kind of business, you can't beat a project like that. That is fantastic science, and also has the potential to really revolutionize not only how toxicology and assessment is done, but actually how drug development is done as well. As Ivan said, I come at this actually as a physician first, and then as a geneticist second, and as a drug development third, and as a Johnny-come-lately toxicologist. Although I must say that when I was a doc, I dealt with this problem all the time, and being at Merck, I dealt with this problem again because a lot of the reasons that the projects I worked on failed were because of toxicological reasons, and we were always questioning, well, gosh, what does the animal data really tell us? And so it was a wonderful happenstance that the planets aligned, and we came together with EPA and NIHS a number of years ago to start this Tox21 collaboration here. So Ivan asked me to, as he mentioned before, to not talk in a parochial way, but rather to give you a sense of what these data can do and not do. Probably many of you are relatively unfamiliar with this kind of approach, so I'm glad to answer whatever even very basic questions you have. I find that my academic and government colleagues are less familiar with this approach because it simply hasn't been present outside the venue of pharma traditionally. So I'm going to use Tox21 data and our approaches sort of as an illustrator, but I'll try to bring out some general points. So, okay, so as you know, Tox21 is made up of 3 organizations, and you'll hear from Richard Judson later from EPA, and you heard from Chris Poirier already from NTP and AHS. And of all the things that Tox21 does, really all I'm going to talk about is right here, is the ultra high throughput testing that we do at our place, and then the mid to high throughput testing that we also do, and then I think Richard will talk about some of that work as well through the ToxCast program. And this is the way at least I like to think about this, that we started out with these data over here, and most of the data that we've been using to do toxicological risk assessment has been based on these data historically, and as a result there's a huge amount of data from EPA and NTP historically, and I think Richard will talk about some of that. What we really want to know is over here, human in vivo. We really want to predict that. And so how do we get from here to here? Well, unfortunately, we've tried to go directly from here to here, and we're all familiar with the hazards of doing that because happily rodents are not humans, or humans are not rodents, or what have you. At least most humans don't behave like rodents. Some probably do. But what we're able to look at in Tox21 is, of course, in vitro data, either rodent cell lines, rodent targets, et cetera, or human cell lines, human targets, et cetera. And I must tell you, one of the things, just to bring out one of the controversies that we're dealing with all the time within the program, is to what degree should we generate data that's rodent on rodent cell lines, rodent constructs, rodent targets, because that would allow us to make more direct computational connections or correlations with the rodent in vivo data. We started out with the concept that rodent in vivo and human in vivo are not necessarily directly comparable. So if we take human in vitro data and try to correlate it with the rodent in vivo data, it's probably not going to be the best approach. However, really what we ultimately want to do is not predict rodent in vivo. I mean, we'd like to do that in the short term, of course, as Chris was saying. We really want to be able to prioritize. But we want to make sure also that we focus on these as well. So we're really generating both types of data. These data, of course, are really, from a toxicological standpoint, we don't have and will never have, of course, from environmental chemicals. The only place where these data are present have been gathered in a systematic way is, of course, with drugs, which people will happily and do happily take voluntarily in many different doses over periods of time. And so those data, of course, are required for FDA approval. So those drugs, that drug data is present. We're currently working with FDA, and I actually have a meeting tomorrow or day after tomorrow with FDA to talk to them about joining the MOU, and they're very enthusiastic about doing that. And we're trying to work out issues of data sharing, and there's issues of secrecy, or not from FDA's perspective, but from the perspective that when data on drugs are submitted to FDA, they're done for a specific reason, for drug evaluation, and they can't simply share those data with anyone else because they asked for it. And actually, the data that you're looking for can't even be FOIA-able, Freedom of Information Act, for those of you who are not from Washington. Okay, but we're working very hard on this, and through the efforts of Bob Kavlock at EPA and others, a number of pharmas have actually come up voluntarily and said, well, we'll share some data on failed drugs with you and share you all the animal data, et cetera. So it could be that we'll get some of this data directly from pharma. Okay, so definitions. What I'm supposed to talk about is down here, and so let me give you some definitions about how I talk about screening, and this is really, I think, how most screeners would talk about screening. That by definition, most people talk about high throughput screening as meaning 100,000 tests a day or more. In our place, we do about 500,000 to 1 million tests in a day. So that's high throughput. Medium throughput screening, you might call this where tens of thousands or thousands a day. Tens of thousands, generally what you're talking about is more high image-based or high content screening, like a complex phenotype like neurite outgrowth, et cetera. One can certainly do, and we do this all the time, image-based screening in a high throughput mode, but the images are relatively simple. It's just an issue of data collection and analysis and speed thereof. If you go down the scale of throughput to model organisms, worms, zebrafish, flies, etc., then we can only do thousands a day, if that. And then, of course, the traditional model, which one might call low throughput screening, which is the rodent itself. So the way I think about this, and I think it's a helpful sort of rubric, is that high throughput screening is incredibly valuable because of the T, the throughput. It allows you to test very large numbers of compounds in a very rapid fashion. Just to give you a sense of this, we can test the entire Tox21 collection across an assay in a single afternoon in 15 concentrations. So we can test 2,000 to 3,000 compounds at 15 concentrations in a single afternoon in an assay. Then that's great. It also gives you insight into molecular mechanism if the farther you've become reductionist here. So just imagine the extreme here. This is a picture of a protein, of course. If one has a isolated protein in a tube or in a well, and you add a compound, and the compound does something, it's very likely that compound is interacting with the protein, because that's basically the only thing in the tube it can interact with. So you know a lot about molecular mechanism. However, when you do that, you have taken the protein out of all of its physiological context. So it doesn't have its protein binding cofactors, it doesn't have its cytoplasmic or nuclear milieu, it doesn't have the cellular context, it doesn't have the tissue context, doesn't have the organ context, doesn't have the animal, the organism context, doesn't have metabolism, et cetera, et cetera, et cetera. So this is extremely useful, but you have to take these data for what they are. They are a reductionist representation of what this compound can do in an isolated system. But woe betide you to try to take these data and extrapolate immediately over here. You just can't do that. And I see this mistake actually in drug development all the time. We do assays like this all the time in drug development, and then I will get asked very, very regularly, okay, we've done the screen, when do we go into people? And at that point, I usually leave the room and cry, and then I come back and I feel better. But in toxicology, it's no different. It's exactly the same issue, except toxicology is even harder, because toxicology means every possible thing that could go wrong in an organism. In drug development, we're looking for a very specific outcome. And here, we have to know everything from reproductive tox to neurotox to skin to lung, nerve, et cetera. Okay, so if you move your way back this way, then you get more immediate organismal relevance, but of course, as has been pointed out, your throughput goes way down. So the concept is use high throughput screening for what it's good at, to identify molecular mechanism, to get high throughput, but then be very careful about doing follow-up assays or correlative assays, working your way up the chain of biological complexity before you really make strong conclusions. Okay, so for those who aren't familiar with this, basically the way to think about this is what can you screen? Everyone wants to know about that. If you can fit something into a 1536 well plate. So we do all of our screening in a 1536 well plate. I should have brought one of these, but they're the same footprint as a 96 well plate, except it has 1,536 wells and a total volumes of about 10 microliters, about 1,000 cells on average. So any assay, any testing system which is cell-based or cell-free and will generate light of some sort, we can assay. So the only things we don't do at our place are metazoans, multicelled organisms like this. So the only organism we do is yeast, so anything that's cell-based or below, either image-based or pathway-based or proteins. And we don't do radioactivity either, but that hasn't been a problem because there's lots of alternatives to that. So essentially anything that will generate a light of any wavelength, so fluorescence, luminescence, time-resolved fluorescence, absorbance, anything like that. Okay, so for those of you who want to learn more about this, I would refer you to this site on our website, which is just ncgc.nih.gov. It's a very extensive assay development manual that was put together by a bunch of folks at our place, together with colleagues at Eli Lilly. And it's, for those of you who are molecular biologists, you know about Maniatis, those sorts of current protocols in molecular biology, those kinds of things. This is about a 300-page manual about how to develop an assay, how to QC it, whether you can trust the data, et cetera, et cetera, et cetera. It's very detailed. Okay, so if everybody likes to see robots, so I thought I'd just show you one picture of this. This is one of our major screening systems, but we have about 4 others. This is about the size of a small school bus. It's about half again as big as this room from one end to the other. We can fit about 4 million compounds at this end. There are tissue culture incubators in the middle, cell dispensers and readers down at this end, and each of these big yellow things is taller than me. It's about Ivan's height, and these big yellow arms just act like postdocs or technicians. They just transfer plates back and forth to each other all the time, all day, but they don't complain, et cetera. Much higher throughput. So we can run through, and we do every week. We run about 4 million tests a week on this system. So the readers, I don't want to get too weenie on you here, but just to say that there's a number of readers which are either CCD based or PMT based, which are look at the total signal coming out of the well, or these are more high content readers, which are laser scanning cytometry or true microscopic imaging. We also have ability to do rapid kinetics, sort of like a flip or calcium imaging, if you're familiar with that. Okay, so I want to make a distinction, and those of you who've seen me talk about this before, pardon the repetition, but I think it's important. How high-throughput screening is done in drug development is the following, and I want to make a distinction between this and how you have to do this for toxicology. So yeah, first thing that you do in drug development is you identify a target. Say if you're going to identify a HMG-CoA reductase or something for the statins, and then one will create a testing system, or otherwise known as an assay. Assay, as you know, just means try. So when it has a cellular or biochemical phenotype that you're looking at. And then what you do is you're testing lots of chemicals that have no known activity. As a matter of fact, they are intended to have very, very narrow activity. And so most of these are novel synthetic compounds that have never been tested in any biological system before for an effect on a target. And the prior probability of activity is extremely low. So the average hit rate is 1%, 0.1%, something like that, because one's looking for a monospecific compound and one screens for that. And then what you do is you spend usually somewhere between 3 and 8 years making modifications to these chemicals from what came out of the screen in order to make something which is safe and efficacious in an animal, and then you keep making modifications to make it safe and effective in a person. So there's a couple of distinctions here. First of all, in toxicology, these compounds may not have known activity. Some of them do, but they have suspected activity. There's some reason why they're on the list. And also, we're not doing this. We're not doing medicinal chemistry to try to improve the toxicity of our compounds. So it's other big difference. So qHTS for toxicology. So you're taking chemicals with some sort of feared or known or suspected toxicity in humans. There's some reason for concern here. And then one goes backwards to identify the Mel Anderson Memorial Toxicity pathways and cellular phenotypes. And then you do the same thing. You either obtain or you create a testing system to test these things, just like you did in drug development. And then, of course, you test the chemicals for effect on the assays. But there's two big differences. One, your prior probability of activity is higher, and secondly, your tolerance for false negatives is much, much lower. With false negatives, you can get away with in drug development, because remember, all you're doing is looking for a couple of compounds that you're going to start a med-chem campaign with. Here, false negatives are unacceptable, so the approach has to change. And then you do computational analysis, synthesis of the HTS results, and then you result in one of two things. At this point, you either, eventually, of course, we want to be able to predict directly in humans, but at this point, we're prioritizing chemicals in animals or categorizing as inactive in this initial assay, subject to another round of testing. But remember, inactive here just means you tested it in one very small node, so it's only as inactive as how many tests you tested it in. Okay, so why is high-throughput screening as conventionally done for drug discovery not suitable for toxicity testing? Well, the first two reasons are this, that single concentration, so just go back a second. High-throughput screening, as most people mean it, means a single, fairly randomly chosen concentration, generally 10 micromolar, sometimes 3 micromolar, 2 micromolar, but it's a concentration chosen for convenience, and it's tested at a single concentration, and then because the prior probability is low of any activity, the vast majority of the compounds have no activity, so they have zero activity. And then what one does is plot the activity of these compounds and calculate statistical deviation from a mean of 0, where most of the compounds live. And so one can draw a 3 standard deviation cutoff of activity or 6 standard deviation cutoff of what have you, and you might imagine as you do that, if you're more stringent, then your false positives go down, but your false negatives go up. The usual sorts of statistical things one can do. So I don't need to tell this crowd that this is a really bad idea for doing toxicology because the false positive rate and the false negative rate are extraordinarily high. Prior probability is low. You can't, as a result, in my opinion, you can't reliably bank or computationally synthesize these results because if you take results like this and you try to compute on them, you're going to get garbage. And so you have to be very careful with what you actually put into your computational algorithms. And as we said, the purpose is different. So it's not that screening for drug development is bad or good or whatever. It's just different. It's a different approach. So there's different reasons. So we have to do it differently. Okay, so this is our patron saint, of course, who first pointed out that dose makes the poison. So this is as true with screening as it is in animal tox or human tox, for that matter. And so we took a cue off of recent papers from Paracelsus to suggest that we should do screening in dose response. So that's what we do. It's something called quantitative high-throughput screening. And there's a paper in PNAS 3 years ago or something like that now that describes this. But I describe this where conventional high-throughput screening done at a single concentration. What qHTS does is what all toxicologists have always done, is think about things in terms of dose response, which, believe it or not, is not, well, you probably would realize this, but if you get outside of the realm of toxicology, most people, including molecular biologists, they think in a bifid world, positive, negative, 0, 1. It's a sort of digital world. But biology is not digital. Biology is quantitative. So this is the general range that we screen in. It's one of the reasons we do 1536 well placed, and what you get is a concentration response curve for every compound. So instead of getting a statistical deviation from 0, you get activity profiles for all compounds, dramatically reduce false positive, false negative. So I said, don't worry too much about the throughput. The numbers are huge. So in your discussions and in our discussions, screening throughput is not limiting. Just put that into your head, and as soon as you realize that screening throughput is not limiting, then it fundamentally changes how you think about what questions you can ask. So screening is now an experiment. It's not something that you lead up to for your whole life and then spend your whole life trying to deconvolute the results. It's something that you do in an afternoon, and you can repeat it the next afternoon if you want to. You can test thousands of compounds in dose response in a single afternoon and do it over and over and over and over again. So I think will probably be obvious, you have to do this in order to cover all the kinds of phenotypes and responses that you're interested in looking for. So again, I'm not going to go into detail, but just to show you that this will warm the cockles of your heart, I hope that we classify compounds according to their curve class down here in conventional pharmacological terms, 1, 2, 3, 4 curves, and these curves at this end are more, the compounds that produce these curves are more robust, and these are more subject to false positives or false negatives. As a result of this, in order to analyze these kinds of data, we now have in our whole collections about 400,000 compounds in the whole collection, of which Tox21 is currently about 3,000, but it'll be about 10,000 by the end of this year. And we've done about 300 assays at this point. So one can look dynamically across all the assays and all the compounds and all of the assays that a particular compound hit in, or all the chemicals that hit on a particular assay. And in this case, cool colors means it inhibits the target, red means it activated the target. And if you click on any of these, it'll bring up the dose response curve off the robot and the structure, et cetera, et cetera. And you can cluster these by biological similarity or chemical similarity and that kind of stuff. So this is the kind of thing that you need in order to analyze these kinds of data. And you can look at it in terms of structures and curves, or in terms of just colors as well. Okay, so just to show you, we did our homework when we started this, and we started working with initially NTP and IHS, and then about a year later with EPA back in 2004, 2005. We looked at intra-experiment and inter-experimental reproducibility, and it was quite good. Just to show you the kind of data that's produced, what you can do is look in terms of potency range, whatever potency range you want, and what kind of clear classification you're looking at here. And of course, most of them are inactive, and then you have actives and what are annoyingly called inconclusives. This is a reality of doing any experiment, of course, if one has to put results in a bin. And so we spent a lot of time trying to figure out what inconclusive means and how heavily to rely on them. If you haven't seen this, we published this proof of principle in EHP about a year and a half ago. I'm a final author if you haven't seen that. And then we published an initial computational analysis of these data also about a year ago in Chemical Research and Toxicology that was looking at trying to discern how many different molecular mechanisms were there in this data set. So if you want to look at that. Okay, so to give you an idea of the toxicology assays we've screened so far, so they range from general toxicity, cytotox, apoptosis, membrane integrity, mitochondrial tox, gene tox, these bunch of tox pathways here, and a large number of molecular targets, generally in reporter assays, and you can see the variety here. So it's a very large variety of assays. And this is the kind of immediately interpretable data you get, which I'm not going to go into because you're going to be suffering through a lot of heat maps for the next 2 days. So just to say that you get results which you can then cluster and then try to make sense of. Okay, so this is so that's the good news. What's the bad news? And Ivan asked us all to focus on this, and this really is important for a variety of reasons. Limitations. As I said at the beginning, these assays are necessarily simple and reductionist. They're that on purpose. They're frequently artificial, like in reporter cell lines, where it takes a promoter and hooks it up to a reporter. Takes a promoter and hooks it up to a reporter like luciferase or beta-lactamase. The assays really only usually report on one aspect of a signaling pathway. So if one has a TGF-beta pathway or something like that, usually one's stimulating with one kind of agonist, so there's many other ways to enter into that pathway, and you're only going to be looking at one part of it. They may be precise, i.e. experimentally robust, and we actually don't do any screens until we can demonstrate to ourselves that they are, but not biologically accurate. And this difference between precision and accuracy is really critical. Not all in vitro assays are suitable for high-throughput screening. And it's one of the reasons why you sometimes kick these assays back to the medium throughput screening sort of setup. Not all substances can be tested in vitro, the volatiles, for instance. Generally, and this gets a little bit hairy here, so stick with me with the terms. They're generally a little bit limited to a cell-autonomous effect of a parent compound. What does that mean? Parent compound you can imagine, so in these systems there's no metabolism. We're trying to do co-culture experiments with hepatocytes, et cetera, but you can imagine the limitations of that at this point. Cell autonomous means that there is the responding cell type is the one cell type in the dish. So if you have, in a normal physiological situation, if you have an effect on one cell type that then signals a different cell type to do something, but A is required to have B, you're not going to see that in an assay like this. Metabolism, of course, huge issue that we're working on. Exposure, genetic heterogeneity. Ivan and our group are talking about an approach to this that we've done a little bit on. And this is how Ray likes to put it, to just constantly beat into folks that a gene is not a pathway is not a cell is not an organ is not an animal, etc. Okay, so how do you address these limitations? And this is going to sound funny coming from me, but folks who work in this field, if they're honest, and not all are, but if they are, they will tell you this. You must view all these results with a high index of suspicion. The mantra in our place, which I promulgate to everyone, for every assay, is all results are artifacts until proven otherwise. And when you do screening like this, when you test millions of compounds, the chances are you're going to get a lot of artifacts. So you have to, if you go into the experiment with this in mind, it doesn't invalidate the approach. It just gives you a proper perspective of how much you can trust these data. So in order to do that, you have to confirm results in other assays of the same phenomenon. So say in the simplest test, if you're looking at a gene reporter assay, you look at one with a luciferase readout, one with a beta-lactamase readout, and if you get the same result, that makes you happy. Or you go to lower throughput, more physiological systems, and that's what you do in drug development every day, as I started with. Test the same pathway, readout in multiple different assays. I mentioned that move from cell lines, like 293 cells, et cetera, to primary cells, which we're doing, incorporating metabolism, which we're trying to do, and determine responses in cells from different genetic backgrounds, which we're also starting to do. So needs, how can you help us? Determine which, we really do need help, and we're hoping from this meeting we'll get some guidance on this, because this really is it's a very hard problem, and we need everyone's input on this. Determine what cell types to use. I mentioned the human to rodent, primary, stem cell derived. We're working on all these things, but which ones are the most useful? To develop a comprehensive battery of pathways and phenotypic assays. Really, one is ultimately looking at the entire melange of pathways and networks within a mammalian cell that one needs to query, in the concept that if that physiological pathway is important, if you perturb it, you will get a bad effect, otherwise known as toxicology. So you really have to have a very broad-based approach and be able to look across all assays and phenotypic assays. Incorporating metabolism, we figure out how to do that. Secondary assays I mentioned. This is something which I also mentioned just briefly, obtaining in vitro experimental animal and human data from the pharmaceutical industry. And this is what you're going to talk about, developing meta-analyses, so I'm not going to spend any time doing this. And then ultimately, this is where we're trying to get. But we're really at this point, at this stage. So the take-home messages, the vision in the NRC report has really been implemented. It's now in place. We've done many, many screens and continue to do them. And Richard's going to talk about what ToxCast is doing well. I think I've convinced you, I hope, that HTS is invaluable, but it's only the start. And the CompTox community is an essential partner for a number of reasons. It's not just the meta-analyses, which we spend a lot of time thinking about, but assessing individual screens. Each result yields over 200,000 results. It is impossible for a person to analyze those results. It has to be done with computers. Comparing results of similarly focused screens to try to go from this issue of precision to accuracy, and then all these cross-pathway meta-analyses. So here's my email. If you have questions, Bob is here as well. I don't think Ray's here today, but if you have any other questions, just email me, and I'm glad to answer any questions now if we have time.
So this is a very nice talk, and I learned a lot. So my question for you is that, so is the NCGC data publicly available?
Oh yeah, we are public. Yeah. So I get about 2 or 3 emails or calls a week from people saying, hey, I've got this great target or this phenotype, and I'm looking for a small molecule modulator of, or I've got this tox assay that we want to run, and what do you think? And so, yeah, we have about 100 ongoing collaborations. So yeah, by all means. we're always looking for good ideas.
Effects of chemical exposures. So what is your opinion or your view on using this type of mechanism to test multiple chemicals at the same time?
Yeah, it's a great question. The puck in the room would say that frequently we're testing many compounds in a well, even if we don't realize we are, because compounds tend to degrade and they may not be pure from the beginning. So we probably are testing mixtures without actually meaning to test mixtures. But from our standpoint, mixtures are no different from a single compound. From a screening perspective, we actually do this very frequently in natural product extracts that we screen. Either crude ones or partially fractionated ones. And so the issue there is, of course, one of deconvolution. If you get an effect, what's it due to? Is it really is? If it truly is additive, is it synergistic? Does it require both? That kind of thing. But mechanistically, it's absolutely no different. It's a single compound from a mixture is really the same.